Hello, and thank you for joining us. I'm Gwen Taylor, Senior Editor with Current Protocols at John Wiley & Sons, and I'm here today to introduce a webinar entitled Stem Cell and Reprogramming Research Methods, Practical Considerations for Studies of Disease, including Culture Methods and Genetic Manipulation. This webinar is being co-sponsored by Current Protocols and R&D Systems. An industry leader for over 25 years, R&D Systems develops and manufactures high-quality reagents, bioactive proteins, antibodies, and related tools for the biotechnology research market. R&D Systems products assist scientists across disciplines and are manufactured under strict standards to achieve the highest level of performance and consistency. Among its offerings, R&D Systems provides specialized media and complete kits for the expansion, characterization, and differentiation of distinct stem cell subtypes. Current Protocols is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 15,000 protocols, Current Protocols is part of Wiley Publishers. We have allotted 1 hour for today's program, but our speaker has agreed to stay on a bit longer to answer as many of your questions as possible. You can submit your questions throughout the event by typing them into the Q&A box at the bottom right side of your screen. The webinar will be recorded and available for viewing in the next few days. We will send each of you an email with details on how to access the recorded webinar along with a PDF of the slides. Now it is my pleasure to introduce today's speaker. Dr. Kevin Eggan is a Howard Hughes Medical Institute investigator at the Harvard Stem Cell Institute. He came to Harvard University in 2003 as a junior fellow and then became an assistant professor of molecular and cellular biology at the Stem Cell Institute in 2005. In 2012, he became a tenured professor of stem cell and regenerative biology. Among other high-profile awards for creativity and productivity, Dr. Eggan received the MacArthur Foundation Genius Grant in 2006, and in 2009 was selected as one of 50 Howard Hughes Medical Institute early career scientists who received 6 years of dedicated support to conduct transformative research. His current research focuses on applying the knowledge gained in stem cell biology to studying the mechanisms underlying ALS and discovering new therapeutic targets. So let's begin with the very warm welcome to you, Dr. Eggan.
Gwen, thanks so much for the invitation to be here today, and thanks also to Current Protocols and R&D Systems for sponsoring this event. It'll be a real pleasure to tell the listeners today about our research and about our efforts to try to tame stem cell biology to tell us something about human disease. And indeed, that's what I hope to do for the next 30 to 40 minutes, is to give a high-level overview with an occasional deep dive into the types of methods and approaches that my laboratory at Harvard has been using to try to really accomplish this goal. So I think it's really become clear to everyone over the last few years that in principle, stem cell and reprogramming biology together offer an unprecedented opportunity to access large quantities of neurons that we've never been able to obtain before from the human body. And why is that important, and why is it timely now? Well, it's because simultaneous to this advance in stem cell research, really a revolution in human genomics has delivered to us an unprecedented wealth of genetic information and a number of genetic variants for really many different diseases, but particularly in the neurological disease and the neuropsychiatric disease space that underlie the human neurobiology of disease. So essentially, a real opportunity has presented itself at the same time that a real problem has presented itself, and that is that, namely, for the first time we can make these neurons, and we have a real need to try to understand how the genetic mutations that we're coming to appreciate cause these conditions change the fundamental properties of those neurons. Now, what I think I truly and earnestly believe is that over the next few years, that will mean that we'll be able to basically take these stem cell-derived neurons and use them to functionalize these genetic discoveries, to really take the information about these genetic variants and turn them into some fundamental understanding about how they affect neuronal function. And I really think the remarks that I will give today, although they'll be drawing upon our experience in studying the neurobiology of disease, are really general lessons that anyone could apply when attempting to use stem cells and pluripotent stem cells to understand diseases of any kind. I think they're really very general efforts and general approaches that are useful for people who are trying to use stem cell biology to ask genetic questions. Now, I think that the heart of this and the methods that I'll talk today are really strategies and methodologies that we and others have developed, which can help researchers deal with the various forms of reproducibility problems, forms of variation that crop up in the use of stem cell biology to ask these kinds of questions. And because there is so much potential of stem cell biology, because these cells are so powerful and they do recapitulate many aspects of development, it means that as an investigator embarks on these types of experiments, really the sky is the limit on the number of approaches that one could take, but also the variety of outcomes that can occur in a particular experiment. And that can lead to real problems in reproducibility from investigator to investigator and laboratory to laboratory. So how can we overcome some of these problems, and what are the strategies that we take to do that? That'll be what I try to really address today, as well as, I hope, giving some thoughts into the future about how thoughtful individuals might be able to better understand disease by taking advantage of these methods. So I think another important concept, another really important piece of background information, which is inherent to my introductory comments, is that really this change in our understanding of the genetic architecture of disease is beginning to change the way that we think about diagnosing disease, studying disease, and treating it. And that really is beginning to play very nicely into the hands of individuals who are using stem cell biology to study these conditions. So in the past, there was a very hierarchical means of thinking about disease-related studies, and you can see that in panel A here in the left-hand portion of my slide. In classic diagnostic terms, diseases were more or less classified into subgroups. So, for instance, you might think of an immunological versus a neurobiological disease, and then, for instance, perhaps a degenerative condition rather than a cancer of the brain. And then, based on very specific clinical syndromes or clinical symptoms, a neurodegenerative disease might be classified as, say, Alzheimer's, Parkinson's disease, or ALS, which we'll talk more about. But I think now we're really beginning to realize that perhaps if we're going to effectively treat diseases, we need to instead begin to think about classifying them genetically and using that information to our benefit to try to understand whether or not people get disease for the same reasons, or whether or not there are very many mechanisms that could lead to that particular disease state. That's very important for thinking about how we develop therapeutics and understanding the mechanisms that lead to those particular diseases. So, for instance, it's easy to think about a monogenic disease where one particular mutation, like in this panel B, lead to a homogeneous group of outcomes or phenotypes that might all fall into this single box. In contrast, here in panel C, you could see an example of something that might have been thought of as a single disease, but really is underlied by these 4 different mutations, which all act through different mechanisms to cause clinical phenotypes which are all very similar to each other, but might be modestly distinct. So, for instance, maybe ALS is a good example of this type of disease where the phenotypes that patients have are not exactly identical, but they are very similar and they fall into one particular class of neuromuscular disease. But we now know that there are many different mutations which can act with high penetrance to cause these problems. And then, of course, there may be cases like D, where indeed there are many mutations that might act together and have to actually act in concert to lead towards one particular phenotype. You might think of this as a more complex genetic condition. So it may be, although we're not entirely sure, that schizophrenia, for example, as a psychiatric disorder, behaves in this way, where many different common genetic variants, which we may all carry a few of, but schizophrenics have a larger number of these variants, collaborate somehow through a series of intersecting pathways to cause us to get sick. So this sort of approach or thought process for dissecting disease until now has been a very difficult thing to address because as the different genetic variants, denoted by little dots here on the right-hand side, become more and more common, our ability to keep up with these dots by making new animal models that could read out the effects of those mutations just can't keep up with the rate at which we are learning about those variants from genome sequencing. And so in addition to the ability to really study human neurons, I think stem cell biology is going to offer us an opportunity to really keep up with the proliferation and the appearance of these new genetic variants and begin to allow us to understand whether or not particular diseases fall into one of these types of classes. And I think understanding this is really paramount and an initial first step that's required for understanding how we would think about treating them therapeutically. So for the last several years, my laboratory has really been focused on really understanding this underlying genetic architecture of disease, primarily through studies of ALS. And we've been doing this using induced pluripotent stem cell technology combined with directed differentiation strategies, which I'll spend a good deal of time talking about. So in collaboration with our clinical partners, we've been deriving skin punch biopsies and skin fibroblasts from patients with ALS, both familial forms of the disease as well as sporadic forms of the disease, and then using iPS cell reprogramming technologies to reprogram those fibroblasts into patient-specific stem cell lines. We then use in vitro directed differentiation technologies to make normally unobtainable populations of neurons that we can use for disease modeling studies. Now, I'd like to pause here and just say a few words about this process. From my perspective, fibroblasts are a useful population of cells to use for these types of studies, both because they're easy for trainees and technical staff to grow in the lab, because they represent a population of cells that can be used for other studies besides stem cell studies, and because, for instance, they're useful for other approaches for making previously unobtainable cell types. For instance, for trans-differentiation studies where one might circumnavigate the lower half of this circle and just cut straight across the center from fibroblast to neuron. That being said, I don't think there's anything wrong with using other types of donor cell types, and I think excellent studies are moving forward with the use of blood and other types of cell types as keratinocytes. It's not clear to me that one type of somatic cell has really emerged as a sort of prime example of the cell type that one should use for these reprogramming studies. So I think many things are possible. With respect to direct reprogramming, my lab still uses many tried and true iPS cell lines that were produced by retroviral reprogramming using Shinya Yamanaka's reprogramming factors, such as KLF4, SOX2, OCT4, and in some cases MYC. Many of these lines, if very carefully characterized, are indeed very, very useful. That being said, this is an area where, again, there's been a good deal of technology development in recent years, and it's not clear to me that that any one of these methods is necessarily superior to the others. I think in the face of the development of non-integrating reprogramming strategies that utilize technologies like Derek Rossi's modified RNAs or Sendai virus-mediated reprogramming, I think for me the considerations here at a research level are primarily practical. What can your laboratory do? What can you successfully execute on? What sort of technologies work best in your laboratory? But whatever that is, try to, in the course of your disease-related studies, stick to a singular method for making your ES cells or your iPS cells to try to capture reproducibility at this level. Once iPS cells are made, I'll spend a few moments talking in greater detail about actually directed differentiation of those cell types into, in this case, spinal motor neuron, which are the cell types we've been interested in for studying ALS. And then I'll, instead of talking about cell therapy, really focus on the use of these cells in disease modeling. And really, it's in this second half of the circle here that I'll spend most of my efforts and time, because I think many different groups have talked about details on the right-hand side, and I think that most of the questions I receive over the years really revolve around how should we best tackle in vitro differentiation in disease modeling. So how do we think about making motor neurons from induced pluripotent stem cells and embryonic stem cells? And if one peruses the literature, there are now many papers on the directed differentiation of stem cells into different neural types. Here I'm showing you a protocol that was developed in collaboration with Chris Henderson and Henrik Wichterle's laboratories at Columbia University that we've worked on over the years. One thing that I would really point out before I dive into the detailed description of this is that standardization in these types of experiments is absolutely critical. Our labs and those of Chris and Hennik's spent hours on the phone talking about exactly the details of these protocols, when things would be done, exactly what research products would be used from which companies, so that we could standardize these activities from laboratory to laboratory. This has been absolutely essential for cross-comparison of data between the various laboratories. Without it, we found that the actual neurons that we produce change sufficiently that it's very difficult to say whether or not a particular piece of data and its difference from laboratory to laboratory is the result of some real biological results or some technical aspect of the procedure which has been modified. So one thing that I really urge everyone to do is to get together with their colleagues in the particular field that they're working on and try to come to some standardization to the best of their ability with respect to how they're thinking about making a class of cell. In this case, for directed differentiation of motor neurons, we've arrived at this 24-day protocol for producing lower or spinal motor neurons, which basically revolves around two distinct, I would say, fundamental pieces of biology. First is a discovery that was made by Lawrence Studer's laboratory, in which they discovered that by inhibiting both arms of the SMAD signaling pathway in pluripotent stem cells, that this induced a very efficient form of neuralization. And there's an excellent paper in Nature Biotechnology by Chambers et al. From his lab that describes this development. But basically, treatment of embryonic stem cells or iPS cells with two small molecules that inhibit both arms of the SMAD signaling pathway, this FP compound and dorsomorphin, basically neuralize cells. We then switch the cells out of the self-renewing M-teaser media formulation to knock out serum replacement medium, and then into a neural induction media, which includes things like amino acid supplements, neurotrophic support, in this case BDNF, and then we at that time also rely on the second, I would say, really fundamental biological concept behind this directed differentiation strategy, which was actually a discovery that Henrik Wichterle made while he was a postdoc in Tom Jessel's laboratory, and that is that by exposing those differentiating stem cell lines to retinoids, in this case in the form of retinoic acid and sonic hedgehog, that takes those cells and patterns them to the part of the embryo or the equivalent of the part of the embryo where spinal motor neurons develop. So retinoids have a dorsalizing effect, sonic hedgehog has a ventralizing effect, and that's enough to take these neural progenitors and give them the perspective that they need to become spinal motor neurons. So this is a 24-day protocol, and really in all of our disease modeling experiments, we think of this as sort of day zero of a motor neuron's life. And so throughout the rest of my slides and diagrams, I'll occasionally fall back on timelines for disease modeling. And I think the important thing to keep in mind is that time zero of those timelines is really this 24th day of differentiation. So that is to say that when thinking about these disease modeling experiments, one has to spend a good deal of time still making a particular type of cell that would be involved in a disease modeling experiment. And then we found that often the phenotypes that we're interested in studying take an additional portion of time to arise. And so one has to take that into account. And many of these experiments, indeed, are as many as 2 or 3 months in total length. So one thing that I think is really important to keep in mind before embarking on studies to produce a potential class of cell is, how will I know I have that cell when I produce it? What are the fundamental characteristics of a given differentiated cell type that I could actually test and assay that would give me confidence that I had really produced it? And although it's true that many cell types are defined by the cohort of transcription factors that they express, this can leave one a little bit cold functionally with respect to being confident that one has indeed produced the cell type, given the recycling of transcription factors from cell type to cell type, and our generally limited ability to be able to look at a large number of such things at the same time. So particularly with neuronal classes and the great diversity of neuronal classes that exist, I think that this is an issue of fundamental concern. How can we be sure that we're really producing a given neural cell type? Well, one reason to be interested in studying spinal motor neurons and ALS is that spinal motor neurons are very unusual neurons, and they have very unique properties which are not found in other neuronal types. For instance, they send their projections out of the nervous system into the periphery, and there they make a direct synaptic connection with muscle cells. And here you can actually see a picture from EMBL of the terminal ends of motor axons labeled in green coming into the muscle fibers, which are here in blue. And in red, you can see the acetylcholine receptors in the neuromuscular junction that the muscle cells use to receive acetylcholine from the spinal motor neurons, and through the electrophysiological mediated release of acetylcholine at these NMJs elicit the acetylcholine receptors to be activated and cause the muscle to contract. So here you can see this beautiful overlap between the terminal branching of these motor neuron axons and the neuromuscular junction on the blue muscle fiber. And so one way that we can know that spinal motor neurons are really being produced from this type of protocol is through an experiment such as this that we've been working towards with Victor Reviews in his lab at Halifax. And what you can see is this beautifully terminally differentiated axon and its terminal branching. And here this presynaptic differentiation, which is hovering over these clustered acetylcholine receptors, here stained with alpha-bungarotoxin. And beneath those acetylcholine receptors and near these presynaptic differentiations, you can actually see signs of recycling vesicles there showing that these are active. So these are really spectacularly functional spinal motor neurons that we can make. They make these specialized neuromuscular junctions. And this is just sort of one of the assays that we can do. Another classic assay, which I think shouldn't be underestimated, and it's important even though it's technically challenging, are transplantation assays. So as I mentioned before, motor neurons are special because although their cell bodies basically reside in the ventral portions of the spinal cord here, they send their projections out of the nervous system, which is very unusual for neurons to do, to innervate both axial and limb musculature. And so we can take these labeled motor neurons and transplant them, say, into the chick embryo and see if they'll perform this unusual function of sending out their projections to the limb and axial musculature. And what I hope you can see in this actual picture of data, which is similar to the schematic on the left, is here's the developing chick spinal cord, and you can see that the injected motor neurons here labeled in brown have settled down into the ventral portion of the spinal cord. They're sending their axonal projections both out to the axial limb the limb musculature and the axial supporting musculature here. So not only can we really show that these motor neurons express motor neuron transcription factors, but they really they sort of bark like a motor neuron too in these types of assays. So now that we can actually produce functional motor neurons, what other considerations might we have before we think about using them for studying disease? What are the other types of things that we would really worry about? And I think that a variety of questions about the similarity between iPS cells and ES cells and how they differ from each other and how that might affect disease modeling experiments have been raised, and I want to take a few minutes to speak about that subject. First of all, I think it may very well be true that the average iPS cell line is slightly different from the average ES cell line, but in my opinion and in my experience through studies with Alex Meisner here at Harvard, I think that's really missing a broader and very important point that comes from some of our studies together, and that I think really shape our personal thinking about how these sorts of stem cell lines should be used to look at how genetic variants affect neuronal function, for instance. The experiments that really inform our thinking about this have been published in Cell by Bach and colleagues in 2010, and I just want to describe in a schematic form what I think we found in those experiments. What we did was to derive iPS cell lines from a large number of control individuals and also fall back on a large collection of human embryonic stem cell lines from a variety of sources, but predominantly from lines derived in my lab and Doug Melton's labs here at Harvard. What we did was to take this large panel of iPS cell lines and embryonic stem cell lines and analyze their genome-wide demethylation patterns and transcriptional patterns. What we found was quite interesting. We found that there was a great deal of diversity in the transcriptional profiles of human iPS cell lines here in this more orangey cloud, and also within the population of human ES cell lines in this more purpley cloud. And this variation in transcription, which is denoted here on the x-axis, was mirrored by this variation in demethylation that we observed as well. And there was certainly an average, a center point to this variation, kind of a most average or prototypical ES cell line, and iPS cell line here. And although that was slightly different from the most prototypical ES cell line, what you can see from the schematic of our measures is that there was actually a very considerable amount of overlap between ES cells and iPS cells. So that is to say that although maybe the average is slightly different, there are many iPS cell lines that really behave largely like normal embryonic stem cell lines. And I think that the take-home message from this that's really important is that although there are some differences, there are also many differences between any two iPS cell lines, and there are many differences between any two human embryonic stem cell lines, and those differences are at least as large as this average difference between ES cells and iPS cell lines in principle. So what I think that means is that when you're comparing any two pluripotent stem cell lines to each other, be they two iPS cell lines or two ES cell lines, there are substantial differences between their culture history, their transcriptional programs, their demethylation programs that must be taken into account when doing a carefully controlled experiment. Otherwise, one might inappropriately assign a finding to a genetic variant that is really an accidental result of the differences between these cell lines. And I want to describe what I mean by that in some more detail next. So one functional ramification of that we found that, for instance, could very well lead to confusion in a disease modeling experiment was that when we took the directed differentiation scheme for motor neurons that I just described, and we took these many different stem cell lines and applied that, and even did it again in the 3 independent laboratories, what we found is that each one of those lines had a slightly different propensity for making spinal motor neurons. And that propensity was reproducible from lab to lab. And essentially, I think that, for instance, if one of these lines had happened to have an ALS-causing mutation, it might have been tempting to speculate, for instance, that a line like this one or a line like this one had some change in it that was related to ALS, when it was just simply the overall behavior of that cell line which was causing this phenotype to occur, and that it's really an accidental aspect of the biology of that cell line or its recent culture history, which is making it perform in that way. Okay. So this has real ramifications for thinking about how you design these experiments. So how might we get past this type of problem? How can we move beyond this inherent variation between stem cell lines to learn something meaningful? And obviously, I have opinions about that, or I wouldn't be willing to give this kind of webinar. So let's move on to that. And I'll try to put it into a couple of different contexts and ways that you think about solving the problem that different groups have used. One approach that you can use is to work with a lot of stem cell lines at the same time. So for instance, we could derive many different control iPS cell lines, and then we could go into our patient population and we could derive many different disease-specific stem cell lines, and then differentiate these lines individually en masse into the neuronal type of interest, for instance, spinal motor neurons for studying ALS. And then we could measure some phenotypic component of those cultures. It could be many different things, their physiological properties, their survival over time, maybe their mitochondrial biology, or the length of their processes. And we could make a quantification for the result from each one of those disease lines and each one of those control lines. And that would give us an average measure of the control measurement and an average measurement of the disease measurement. And maybe through that large en masse comparison, we could triangulate into sort of an average or maybe proxy contribution of a disease variant to the phenotype that you're interested in. Now, for any of you who are doing iPS cell research, you know this is extremely difficult to work with this number of cell lines at one given time. Another thing that I'm commonly asked is, well, how many cell lines do I need to work with to be confident about the finding? And I think the answer that I'm forced to give is that it's not clear until one knows what the magnitude of this measurement is, that is to say, what are going to be the error bars on the individual functional measurement that I'll make within a given cell line, and until I begin to do the study and have a guess as to how large this magnitude difference will be between controls and disease, it's very difficult to know. And so I think the number will be directly dependent on the noise and the measurement here and the absolute magnitude of the difference between control and disease. And I think until these sorts of things are discovered, it can be very difficult to make a guess. But in general, the larger this noise is and the smaller this difference is, certainly the more lines one will need to be able to make a convincing argument that they've made a rational discovery. So how can we overcome this problem? The approach that I think is increasingly being taken, and that I'll say a few more words about now, is to take a given cell line and isolate its behavior away from the variant behavior that is found in other cell lines, and to isolate the effects of genetic variants within that cell line by using homologous recombination and gene targeting strategies. So for instance, in this particular case, you could begin by taking a cell line which has a control genotype here in this particular gene with 4 exons, and use a gene targeting strategy to target into that allele this perhaps disease-causing variant, which is here marked in red. And then by doing that, generate an isogenic control, which now is marked by this little this red haze on either side, for direct comparisons with the starting cell line, which would be isolated from some of these problems. So I can give you a concrete example of something that's like this that we've been doing in the laboratory. It's actually sort of the opposite sort of thing, which is also a very useful approach. And in this particular case that we've been working towards, we've been attempting to correct the most common North American mutation in the SOD1 gene, the so-called SOD1A4V mutation, in patient-specific iPS cell lines that we've derived. The approach that we've taken is to actually identify a site-specific nuclease which cuts the genome near the location of this mutation, here denoted by this red bar in exon 1, And in this particular case, it's a zinc finger that we've designed to work at this location. But frankly, one could use most any kind of designer nuclease. Talons could work. Now, CRISPR nucleases are worth considering for this type of experiment. But by inducing a double-strand break with such a site-specific nuclease, what we can do is greatly improve the frequency of homologous recombination at this particular locus. Now here our goal is to take this point mutation and to actually correct it. And so to do that at a high efficiency and with great certainty, we've taken a two-step approach to doing this. So we've made a targeting vector, which has homology arms here and here, flanking exon 1, and then a version of exon 1 in which the mutation has been repaired. We then have an FRT-flanked PGK pyrimyosin selection cassette, and when we take this construct along with an expression vector for the zinc finger, and we co-transfect them into iPS cell lines, we can try to get a homologous recombinant in which the mutation has been fixed and this PGK pyrimyosin cassette has been left behind. So we can obviously isolate this by pyrimyosin selection, and then after screening and checking to make sure that integration has happened correctly, we can then transiently transfect a flip expression vector into the cell line and leave behind only an FRT scar in the genome. And so I think that these sorts of approaches are becoming increasingly doable. And so I think along with that, we should increasingly demand that this kind of certainty is executed to make sure that we can really assign a particular phenotype to a genetic variant rather than other idiosyncratic phenotypes of a given cell line. And the goal, of course, of this is instead of having to use dozens and dozens of cell lines for every experiment one does, to instead be able to generate maybe just one or two control cell lines and diseased counterparts that one could then work with a much more reasonable number of cell lines and neural types to try to hone in on the particular phenotypic variation that one is interested in, and have both a more robust and more reliable disease model, but also importantly, one that takes many fewer graduate student and postdoc hours to execute. So what other problems do we encounter routinely in trying to do these disease modeling experiments besides these fundamental differences between distinct human pluripotent stem cell lines? Well, I would say that if there's one issue that makes our life difficult beyond any other, it is that we know of no directed differentiation strategies, even our motor neuron strategy, which are 100% efficient. And what we found is that during the course of directed differentiation, although it is true that we successfully produce large numbers of spinal motor neurons, we also make lots of other cell types within those cultures, many of which are progenitor cells, eventually glial cell types, or other neuronal subtypes which we have not studied carefully but are reminiscent of those that we would expect to find in the spinal cord. This makes isolating the phenotypic effects of the genetic variants that these cell lines carry very difficult for two reasons. One is that we're trying to understand how the mutations affect the biology of spinal motor neurons, and we have them convened with all these other cell types in the culture, but also because we find that when we do the experiment over and over again, it's often true that the constituency of these cultures varies slightly from experiment to experiment with respect to the relative proportion of these different cell types. Why is that troubling? Well, one reason it's troubling is because one would like to understand the effects of these mutations through things like RNA sequencing of a population like this. And what I can assure you is that the effects on a transcriptional profile, which are caused by having many different cell types in varying amounts from experimental replicate to experimental replicate, are much, much greater than the likely effects of a particular mutation that you would study. And so you might accidentally end up seeing those effects rather than the desired effects of the mutation. So how can one overcome this? One approach is again through genetic modification of stem cell lines to introduce reporter genes which allow you to isolate very specific populations for a more custom analysis of these cultures. So for instance, one can either use homologous recombination or transgenesis or viral methods to introduce GFP under control of either the entire gene or promoters of interest, which only become activated in the specific cell types that one wants to study. I'll give you a specific example of that in a moment. One can then differentiate those cells, and although initially one makes this mixed population of cells, in this particular scenario, for instance, our spinal motor neurons would now be green fluorescent, which you can see here, and that would allow us to use things like flow cytometry to isolate those green motor neurons from the rest of the population. And so now we can have a purified culture of motor neurons for study. And of course, in many cases, we might not have motor neurons alone, but at least we've begun with this pure population, and so we can control how we mix them into other contexts as well for different phenotypic assays. So this has been extremely useful and has really transformed our ability, I would say, to detect the downstream effects of disease-causing mutations in stem cell-derived motor neurons by methods like RNA sequencing. It's really enormously added to the robustness of our experiments. Another reason why this is really important is because in many experiments like these, one often does a time course experiment to understand how a disease affects a given cell type over time. One problem that we've had with the heterogeneous cultures that we produce from stem cells is that they're very dynamic, and that even small numbers of progenitor cells early in cultures like these, which might initially be primarily composed of the cell type of interest, and would early on, here with this pie chart, give you a good deal of signal from the mutation that you're interested in, gradually would be dominated by progenitor cells that continue to grow, whereas of course the neurons that you may do not, and the phenotypic signal that comes from those cells here in blue would gradually take over and obscure any sort of phenotype that one would see. And what we've really found is that by purifying the specific cell type of interest, we've been able to get much more stable phenotypic signatures over time, and have been much more successful in really understanding how the mutations shape changes in these cell populations with time. And I think that really understanding how this, how these neurons evolve and how the mutations change the behavior of the neurons as they mature is going to be a critical issue for the future. So to give you an example of how we solved this for ourselves, because I do think that there are many different ways that one could go about this, we took advantage of the fact that it's well known that the HB9 gene at the level of the spinal cord is very specific to spinal motor neurons. And a transgenic mouse strain had already been made in Tom Jessel's laboratory in which the mouse HB9 promoter was driving GFP. There's a great deal of conservation between the mouse HB9 promoter and the human HB9 promoter. And so we reasoned that if we made a transgene that looked like this one, and we randomly integrated into the genome of a human embryonic stem cell line, that it might report on motor neuron differentiation. And so here you can see this transgenic version of the Hughes3 embryonic stem cell line, which carries this reporter gene from a paper by DiGiorgio and colleagues from our laboratory. And you can see that the ESL line is not green, but as one undergoes days of directed differentiation and goes into timing of motor neuron culture, that these motor neurons do indeed become green, and we can use this marker to flow purify them for further studies. So I'd just like to end with one last thought, which is that I think at this time, people who are studying Parkinson's disease and ALS and other conditions are spending a lot of time thinking about the sensitive cell types to these conditions that they would want to culture and study to understand how mutations affect those cells that are most sensitive in disease. But I think that it's also very useful, potentially, to not only think about making these sensitive cell types, but to also contemplate how would we make cells that are reminiscent of those that are more resistant to disease in those particular patient populations, and to use that information to understand the particular effects of genetic mutations and how they actually cause neurons to get sick. And I can give you one example of this sort of thinking from studies of motor neurons. It's not something that we've implemented yet, but I think it's something that we're excited about for the future, and I'm hoping to inspire thought about. So in ALS, ventral motor neurons are exquisitely degenerating in this condition. So as I mentioned before, ALS is a motor neuron disease. These cells are the ones that are most sensitive to disease, and so are the ones that we've been thinking about making. In contrast, the more dorsal sensory neurons, which are also very large projection neurons, are more resistant to disease, and even though they carry and express the same mutant genes that cause these neurons to degenerate, these do not. And Lawrence Tutor's lab and others, like Clifford Wolf's lab at Children's Hospital, have been working hard on making these types of DRG sensory neurons. And so a really interesting thing would be to ask, what is it that's different about how these cells respond to the disease-causing mutations compared to these cells? So if I can just give you a sense of that, for instance, if we could make both sensory neurons and motor neurons from control and disease cell lines, one, for instance, might in a particular hypothetical experiment think that that motor neurons might not survive as well if they have an ALS-causing mutation, whereas the control sensory neurons might do less poorly. Whereas both control neurons might survive. That's to say that neurons of both types that lack the mutation might survive very well. And if one could really have this kind of carefully composed cell lines, it might lead you to both the identification of genes that, for instance, change in the presence of the mutation in both types of neurons, and also eventually to genes which are only modified by the mutation in these neurons that are most sensitive to disease. This kind of insight would kind of give you a selection for perhaps, or a prioritization for the most important disease-related changes that occur in the most sensitive types, the things that kind of represent the critical path to degeneration that one might want to stop. So I hope that this has been a useful perspective. I hope that you found my comments useful, both in a specific level and a broader level about how we're thinking about approaching these types of problems. I haven't said a lot about the specific assays because I really think that that's limited to the particular questions that you're asking rather than something that's related to this overall platform technology. I really hope you've enjoyed it as much as I have, and I'm very happy to thank Gwen, R&D Systems, Current Protocols, and all of you for your attention, and I'll happily stay on the line and answer questions.
All right. Thank you, Kevin. It's now time for the question and answer session, so if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's go ahead and see what questions have come in so far. The first is it true that subculture methods, whether they be enzymatic or mechanical, can have an impact on the genome of the stem cells and may affect reprogramming? And if so, how?
So I think that's a great question. The best evidence that we have for something like that comes from our studies in female embryonic stem cell lines and iPS cell lines, and there what we found is that the most ESLs and iPS cell lines, female ones, have an inactive X chromosome. However, through a process that's not completely well understood, but is, I'd say a hint of it is described in several papers that have been published in Cell Stem Cell from our lab, Jean Loring's lab, and Shinya Yamanaka's lab. There seems to be a process in which that inactive X chromosome gets reactivated. And that can dramatically affect X-linked DNA methylation and X-linked gene expression. This can subtly change the phenotypes of cells if you're studying non-X-linked alleles, and can have dramatic effects if the mutation that you're studying is X-linked. So you could go to a paper by Mekubat et al. To further learn about that. I think that's the best example that we have of something that from passage to passage can have a substantial effect. I think the direct evidence from passage to passage change is less clear. Instead, I think in those measures there seems to be a bit of an evolution over time in culture on average from a performance perspective. And you could check out the paper by Bach et al. In Cell from 2010 to learn a little bit more about that.
Okay, next question. Is it possible that stem cells that are used for therapy can themselves produce mutations?
So repeat the question one more time.
Is it possible that stem cells used for therapy can themselves induce mutations?
I don't think we have any evidence for that per se. I think that it's, of course, a substantial concern that people have that stem cell derived tissues would have mutations that might increase the risk of cancer. But I don't know that those risks, we have any evidence that those risks go beyond, at this time, what would have for any cultured cell that one would use for a transplant.
All right. Next question. In disease modeling, how do you ensure that the reprogrammed cells have incorporated all the necessary mutations?
The way that we've done that is in the case of specific mutations that we've been going after, when we're, say, using a patient-derived fibroblast or blood cell, we'll simply regenotype the patient we'll regenotype the cell line after we've made it. So we'll use PCR or a sequencing approach to actually make sure the mutations are there. For bigger projects that we're undergoing now, where we may not know exactly what the mutation is that's causing the disease, we've been using sort of more classic genotyping approaches like SNP arrays to keep track of the identity of the fibroblasts and the downstream iPS cell line.
Okay, next question. How do you think about modeling adult-onset degenerative diseases with stem cell-derived neurons that have not undergone the same aging process that is a component of the disease in patients?
Yeah, I think this is a question I get a lot. I think it's an important question, and I think that the way that we've been conceptually thinking about this is the following. Those people who have mutations which, with a high penetrance, that means with a high certainty, cause an adult onset disease, must have something wrong from the very beginning, which is caused by that mutation, which will eventually go on to cause the disease. And so we may not be able to study in those cases exactly the events that are happening in the patients at the time that they get sick, but I think we can ask the important question, how does this mutation that we're interested in change the fundamental biology of those cells from the very beginning? And then by understanding that and the series of changes that happen in those cells, we can get closer and closer over time to what's happening in the patients.
All right, next question. This one's a bit more specific. It says, do you have any suggestions for cryopreservation of iPSCs, both feeder and feeder-free cultures? We've been using fairly standard methodologies for freezing cells. Standard freezing medias plus serum. I would say just simply don't scrimp on the actual number of cells that you're freezing at any given time. Using things like ROCK kinase inhibitors can also help with cell survival in contexts like passaging and freezing. I would generally say that actually perfecting and improving the everyday culture techniques that we use and having media compositions like M-teaser, which dramatically improve how well the iPS cells grow, has helped actually the thawing of cells and freezing more than any process innovation that we have done around freezing the cells themselves. So I'd actually focus more on how you grow the cells and getting better growth conditions, probably rather than obsessing over the exact freezing conditions. All right, next question. How have you practically chosen between the zinc finger, Talon, and CRISPR technologies when carrying out projects?
Yeah, I think this is an important issue too, because I think it's possible to get paralyzed by the evolution and the emergence reproducibly of all these different genome editing technologies. I think it's great to try to use the newest technologies when you start a project, but remember, the point of doing that is not to use the coolest, neatest zinc finger nuclease. It's to actually do a very carefully, genetically controlled experiment. In our lab, we've had success with all three of those different all those different nucleases. We've done a project with zinc fingers. We've done a project with talons. We've done a project with CRISPRs. I think the zinc fingers are much harder to design and to access, and so if you have zinc fingers for a particular locus and you're using them, that's great. I probably wouldn't start a new project with the zinc fingers, given the ease of using talons and CRISPRs. I think if there's a talon binding site and it's easy to design and obtain a talon that's near a region of interest, that's great. That could be useful. Generally speaking, CRISPRs have been easier to employ. If you haven't used something before, it's not so hard to get things together and just with your own molecular biology tools get started. So that's a real advantage. I think there's still a lot to learn about CRISPRs. They seem to be very, very effective, and they cut at the same sites over and over and over again. And so checking for other off-site or on-site effects of the amazing activity of those nucleases will be an important control to do. So in short, to summarize, I would say new projects, think about talons or CRISPRs. If you already have a zinc finger, then it's going to do the job, and you shouldn't just throw it away and repeat your efforts with these new technologies. The old ones are still useful.
All right, next question. Can we say that some neuroreceptor agonists or antagonists can constitute a neuronal growth factor? And in this case, can we use them to develop active compounds to stimulate neural growth?
There's lots of different ways to think about assays for growth factor activity. People have done cell survival assays, plating with and without growth factors. There are now well-established assays for neurite and axon outgrowth that involve live imaging or time-dependent treatments followed by immunostaining. I really think that stem cell-derived neurons are actually great for these kinds of experiments and these kinds of screens. Both to look at the need for growth factors in different conditions with different distinct types of neurons that we've never been able to access before, but also I think you could easily use these approaches to screen small molecules for growth factor replacement activity.
All right, we have time for a couple more questions. This one, if I want to carry out RNA-seq or transcriptional profiling to identify genes changed by a disease mutant, what would be the best way to think about doing this?
I think for us, the way we've been thinking about it is to use the types of reporter strategies that I described and purify these cells to homogeneity early on in the experiment, plate and culture those cells for distinct lengths of time and do a time point assay. And this sort of time course assay allows you to understand first the evolution of changes in transcription over time, and then following that, use these genome editing approaches that I described to make an isogenic version of the line and to repeat the experiment. I think it's critical to try to use these cell purification and genome editing approaches because the local variation in the proportion of distinct cell types that you'll have every time you repeat the experiment and the transcriptional signature which will come from that will swamp out any effect that you would have from the mutation that you're interested in. Also, when we've made transcriptional comparisons between lines, there's enough variation in similar cell types between distinct lines that you may have trouble detecting the effect of your mutation. And so those genome editing approaches are really essential.
All right, final question here. So this is a technical question. Does the transfection method of human iPSCs affect the health of the cells and or homologous recombination? What method is typically used in your lab? So
we've really settled on nucleofection as an approach for doing this. This works great. I don't have any interest in the company that makes those reagents, so I can say that without conflict. It's not a super expensive technology to use relative to others, but for us it's just been essential. The other thing, again, that really helps with the nucleofection is to really use reagents like ROCK kinase inhibitors when passaging the stem cell lines to help with their viability.
All right, we are just about out of time, so I have to conclude the Q&A session. I wanted to tell you that today's webinar has been recorded and will be available for viewing in the next few days. We'll send each of you an email with details on how to access the recorded webinar along with a PDF of the slides and instructions on how to customize and print a certificate of attendance. You can also access the certificate of attendance right now from the Resources tab at the bottom of your screen. So this concludes today's webinar, and we look forward to your attendance at future events from Current Protocols. On behalf of our speaker, Kevin Eggan, and from me, Gwen Taylor, and our sponsor, R&D Systems, we hope you've learned some valuable information, and it's goodbye for now. Thank you so much for attending.